Dear students, in this class we discuss the second method of collecting data, namely experimentation. And experimental design. This is a vast area, and we will consider only the basic elements of experimental design with one or two examples. There are so many complex types of experimentation which we will discuss after some time when we proceed. Now let us see what are the terminologies used in the context of experimental design. One is experimental units. And in an experiment, the individual on which experiment is performed is known as experimental unit. It may be human beings, animals, plants, etc. When the experimental unit is a human being, we use the term subject. When the experiment is performed on human beings, we call it subject. For example, in the folic acid study, with 4,753 women, this 4,753 women are the experimental units. They are the subjects. Then the second terminology used is treatment. Treatment means in the folic acid study, both doses of folic acid, that is 0.8 mg and placebo. Placebo means an inert element, no folic acid, are called treatments. So, the doses of folic acid are known as treatments. Treatment is the doses of folic acid, 0.8 mg and placebo or none is treatment. Generally, each experimental condition is called a treatment, and there will be many experimental conditions. Each experimental condition is a treatment, and there will be many experimental conditions rather than just two. Now, in an experimental design, we have to follow a few principles to ensure that the results are not the result of chance. In experimental designs, we follow some principles to ensure that the results are not simply due to chance. For example, in an agricultural experiment, if an agronomist wishes to study the effectiveness of a new fertilizer or a new seed, he has to ensure that it is not the result of chance. It is the effect of a fertilizer or the effect of a new seed. So to ensure this, three principles are used. Principles used in experimental design. One is control. Control means two or more treatments should be combined. Two or more. In this case, none versus 0.8. 2 or more treatments should be compared. Then only we can establish the effectiveness of the treatment. Second is randomization. Second is randomization. Randomization means experimental units, subjects should be randomly assigned to each group. Here, 2 groups are there, and this 4,753 women should be assigned in both groups randomly to avoid what is known as selection bias. So randomization is required. The third one is replication. Replication means sufficient number of replications should be done. You cannot evaluate the results from 1 or 2 results. You have to repeat this process again and again so that the result is obtained from a vast data set. Then only we can say whether the results are correct or acceptable or not. So these 3 principles should be followed in any experimental design: control, randomization, and replication. Now we consider some of the terminologies associated with the experimental design. Remember this, one of the most widely used common experimental situation is a specified treatment and a placebo. A specified treatment and a placebo. Placebo means an inert and innocuous medical substance. Placebo is something which is known to you. And both are treatments. 0.8 mg, placebo, both are treatments. It is a common experimental situation. And the group receiving the treatment is known as treatment group. And the group receiving the placebo is known as control group. So, treatment group and control group. Treatment group and control group. Also, let us see what are some of the terminologies associated with the experimentation. In the folic acid study, the objective of the researcher is to analyze the effect of folic acid on birth defects. And birth defect classification is the response variable. The response is birth defects, major or minor. Remember this. One group is given 0.8 mg folic acid, the other one is given placebo. So, number of major defects from the group receiving the folic acid, the other receiving placebo. We compare birth defects from the group receiving this and the group not receiving this. It is known as the response. And the daily dose of folic acid is known as the factor. The daily dose of folic acid is known as the factor. And in our example it is 0.8, none. Here we have the data we have presented earlier, the responses associated with both response factor. And in this example, the factor has two levels, 0.8 mg and 0. And these are the possible values of the factor. When there is only one factor, the factor is 0.8 mg and placebo, that is the daily dose. When there is only one factor, as in the folic acid study, treatments are same as levels of the factor. Treatments are same as levels of the factor. Treatments are same as levels of the factor. When we have more than one factor, treatment and levels will be different. In that case, treatment is a combination of different levels of the factor. Different levels of the factor. Combination of different levels. Treatment is here only 0.8 mg and placebo. Only one factor is there. So factor and treatment are same. But when we consider many factors, then treatment and factor are different. Then each treatment will be a combination of different levels that we will see with an example. Now, let us see something about how to design let us now consider one more issue, that is statistical designs. Statistical designs. Statistical designs. Once a treatment is chosen, we have to decide how experimental units are assigned to treatments. Once a treatment is chosen, treatment consists of many factors, combination of factors. We have to decide. Here there is no difference between treatment and factor, both are one and the same thing, as there is only one factor. When there are many factors, treatment is a combination of many factors. Once the treatment is decided, we have to decide how the units are assigned to treatments. For example, in the folic acid study, how this woman is assigned to each treatment, 0.8 mg placebo, like this. Now, let us consider, this is only one treatment and one factor. Let us see another example to clarify the difference between factor and treatment. This is a study based on cactus, a type of plant. 40 cacti is divided randomly into 10 groups of 4 cacti each. 10 groups, 4 in each group. Now, we use what is known as the statistical design known as completely randomized design. Completely randomized design. Now, we use two factors. One is an irrigation regime. And the other one is a polymer. Polymer is a kind of nutrient used for plants. Now, irrigation regimes are of 5 types. One is none. Then light, medium, heavy, very heavy. This factor irrigation has 5 levels. Then polymer has P4, then no P4, no nutrients, then with the P4. Now, in each there are 4 cactus. It is an example of a completely randomized design. One treatment is no water, no P4. Another is light water, no P4, etc., etc. So here factor is different from treatment. Treatment consists of combinations of factors. And this is as 40 cacti are divided randomly into 10 groups of 4 each. It is known as a completely randomized design. Completely randomized design. So here the response variable is, what is the response variable here? The effect of these treatments on cacti growth. The factors are irrigation and polymer. Treatment is combinations of both of these factors, and units, the subjects, not subject here, simply units are cacti on which the experiment is performed. This is, the experimental unit is cacti, response variable is the weight gain or something, factors, as I said, irrigation and P4, and each treatment is a combination of both. And this is an example of a completely randomized design. But this is not the only design possible. We can think about a randomized design. For example, if you consider groups men and women, then we can assign the groups specifying men, women, etc. That is an example of a randomized design only. Then there are various other types of designs that we will discuss later. Anyway, the point is, when we collect experimental data, experimental units should be assigned to various treatments in a scientific way. Then only you will get reliable results. Otherwise, you will not get reliable results. The question is how to assign the subjects or the units is a question of how to design the experiment. Completely randomized design or randomized design. There are other designs like Latin square, etc. So that is something which we will discuss in the context of ANOVA, etc. later. And remember this, in social sciences, observational data collected by means of sample is used to make inferences about population, whereas in natural and physical sciences, both the types of data are used. In natural sciences, observational data is used. Also, data is generated in the lab. Data is generated in a laboratory, in an experimental setup. And how to set up the experiment, that is the statistical design, is important, where some of the terminology is used. Keep these terminologies in mind. We will consider these issues extensively later.